Brilliant. Hello, everybody. This is an event with the Footsteps Festival 2021, which is a year-long festival celebrating living well with pain. The festival was conceived by a group of people who got together on Twitter, and it's a true co-production of people who live with pain, those that care for them, and healthcare professionals. We are very lucky to be supported by the Live Well with Pain website and the Physiotherapy Pain Association, both of whom have great resources, and I highly recommend checking them out. The links will be in chat soon. I'm Nikki Jones, one of the Footsteps Festival organising team and your host for the evening. Tonight's event will have a pacing or a bio break after approximately 45 minutes for 15 minutes, and then we'll be back for what I think is the best bit, and of course, questions and answers. So tonight, it's an immense honour and a privilege to be introducing the Queen of Pain, Professor Irene Tracey. She has a CV that would take me hours to read. Suffice to say, she's top of the neuroscience tree and fascinating and engaging to listen to. She is warden and professor at the University of Oxford and has published over 200 papers. She is also the author of the recently published Ladybird Expert Series book on pain. The details will also be in chat. Basically, you don't get better than this when it comes to learning about pain. So sit back, not forgetting your movement snacks, and enjoy. Irene, thank you, and over to you.
Great. Well, thank you very much, Nikki. That is you've put the pressure on me now with that introduction. I just hope I can deliver, guys, what it is that you're both expecting and what it is that you would like to hear tonight. But it's the honor and pleasure is absolutely all mine. I was just thrilled and delighted to be contacted by Louise and Nikki and Chris. So thank you so much for the invitation. And thanks to you all for spending a very pleasant Sunday evening. I hope you got nice weather where you are. I'm very grateful to you to spend your Sunday evening in my company, and I hope I make it worthwhile. So I'm just going to share my screen now because I have some slides to present that's going to sort of describe some of the ways that we're trying to understand pain better. Can you all see that? I'll get a thumb up maybe from Nikki. Yep, fantastic. Great. So I'm going to take you on a journey through the brain and explain how we've been using some of these fancy brain imaging tools to demystify this very complex experience that we have in everyday situations in acute pain, and then sadly, many of you listening have in your daily lives in terms of chronic pain. Just a few disclosures at the start. I have various roles as Nikki's described, and none of these, I think, present any conflicts of interest, but it's just good for due process to present them at the start of a talk. So let me start by just thinking and describing why it is, as neuroscientists, we want to understand both acute and chronic pain. Well, acute pain, as you know, is the body's warning system. So it's a good thing. Not all pain is bad. So acute pain is a good thing. It tells you that something is hurt, something is damaged, or something is going to be damaged. And so it drives your body's alarm system. Basically, you locate and you discriminate. Is that a heat pain? Is that a mechanical pain? You often have an emotional reaction to that. You attend to where the pain is, and that drives you to make the right decision about what are you going to do, which is often to avoid it, get away from it, and then if it's injured, to seek help and get it relieved. So it's of short duration. But as you well know better than myself, bad or chronic pain, the dark side of pain, is pain that's gone beyond that good warning system. So this is now defined as pain that's still there beyond normal tissue healing time, so about 4 months, persistent, and as you well know, it is of long duration and of devastating impact. There's also just a huge amount of interest for us to understand how we construct this experience of pain, and we're going to go on that journey as to how that is produced in terms of brain mechanisms. So if we go back to the acute pain as one of your really oldest sensory experiences, we still don't really understand how the brain puts it all together, and you have that sensation of, ouch, you know, I've just stubbed my big toe, that really hurts, or I've just burnt my finger in, you know, on a hot pan. And so how we actually put that together is still a mystery and still a really major outstanding question in neuroscience. So a lot of neuroscience, we're trying to understand how the brain constructs how we see and how we hear and how we smell and how we taste. And we've got good knowledge about that. But pain, considering how important it is, we're still on that journey of trying to just understand it from a basic neuroscience perspective. So beyond the huge clinical relevance of understanding, again, what's going behind the scenes in terms of mechanisms of chronic pain, there's just some really interesting and fascinating neuroscience to understand what pain is and how it's constructed in the human brain. And that's, again, what drives many of us as what we call translational scientists, is to understand the blue sky discovery side of pain, and then as well translate that, hopefully, for patient benefit. So pain is, again, as I've said, one of our oldest sensory and emotional experience, and we share it across the animal kingdom. Just here on the left is a little sea slug at the bottom of the sea. And so right from the most simple organism through to complex humans, we all share this experience of being able to detect things in the environment that are going to hurt us, or are predatory or are threatening. And this is something that, again, we all know is very difficult to understand, because the only way we can really understand somebody else's pain is to try and infer from their behavior or from how they describe it what might be going on in terms of the mechanisms and the physiology. And this is really, really challenging. And this leads to lots of challenges for us both, you know, as a scientific level and then at a clinical level, in terms of being able to understand why somebody's pain is a particular way. Is that treatment working? What is the right treatment to give you? And again, this sort of pattern matching of how one infers and reads into you what might be going on is where I think some new ways that we can look at it hopefully will help and improve our understanding of pain, and less rely on just that inference and that reliance on just matching what my pain experience might be like to your pain experience. So again, acute pain, great warning system, drives action. You attend, you remember the things that hurt you and you avoid them in the future. It's a great teaching signal. And then chronic pain now we think of as a disease in its own right. So if you're not aware of this, this is a very important, quite landmark change in how we're viewing chronic pain. For the very first time, it's now being 7 conditions are being defined as primary chronic pain conditions. And what does that mean? That means that rather than being a symptom of something else, and so treating the something else where there's a symptom of pain associated with it, chronic pain in its own state is defined across 7 different conditions as a disease in its own right. And that's really important, because that means that the focus of what it is that we need to look at and search for and discover and then block and stop is the mechanisms of the disease of chronic pain, rather than the thing that caused the symptom of chronic pain. So it's quite a subtle thing, but it's profoundly important. And again, I can go into that in a little bit more detail in the questions, but it's a really exciting and very important thing, I think, for chronic pain sufferers and for us as people researching it, that this in the new classification of diseases has been put there for the first time. As you well know, 1 in 5 people suffer, more prevalent in women and elderly. There's a lot of comorbid problems with chronic pain in terms of mood changes, anxiety and depression. Obviously, there's impact on lifestyles. I don't need to explain that to you. As you know, we tend to put 3 different sort of categories of chronic pain: nerve injury or neuropathic, nociceptive or inflammatory, and then idiopathic or functional. And whilst that's helpful for coding and for maybe allocating the person to the right type of medical speciality, I think it is fair to say that the more we understand the disease and the mechanisms of chronic pain, we know that they're shared across these groupings. And so whilst it's helpful, I think the understanding, again, of what is explaining somebody's particular chronic pain state, we now have a more shared understanding that there's elements of things that are in the nociceptive inflammatory that are also in the neuropathic. There's bits in the neuropathic that are also in the nociceptive inflammatory. So it doesn't sit with these siloed boundaries. There's a lot of underpinning shared foundational changes that, again, we are increasingly being able to understand. It's hugely costly. In the USA, it's now deemed to cost, in terms of the management and treatment of chronic pain, but also people not being able to go to work. And so loss to the economy is about $600 billion. In Europe, it's about 200 billion euros. So it's a huge and devastating impact. And that's, you know, got the politicians interested and listening. And that's also a necessary thing, because that helps unlock opportunities for better, again, patient management, better access to funds, better access to therapies, and better access for research grants in order to try and, again, improve our understanding. And it's defined as one of the largest medical health problems now in that developed world. So a few examples of different types of nociceptive inflammatory, neuropathic or idiopathic, just illustrated here. But again, you're more expert than I, for those of you who maybe have conditions that are primarily in these different groupings here. I don't need to tell you about the opioid epidemic in the USA. less to say that it is a major problem. It's a devastating issue for, again, young Americans in terms of losing their lives. 134 young people in their early 20s and 30s dying every day from opioid overdose. It's a very complex phenomenon, and largely this very sad statistic here in terms of young people dying is due to the very downstream effects of generally having too much opioids around in the system and not necessarily being used appropriately in terms of chronic pain management, leading to a culture of opioids. And then, of course, inevitably, sadly, what happens is that synthetic combinations are produced, and street fentanyl being out on the street and being incredibly lethal. And for the students that I teach here in Oxford, I show them just a little vial of what it would be, to take in terms of heroin to kill you. And there's a couple of sort of millimeters of white powder in the base of a test tube, and then show them what it would be if you had the synthetic fentanyl. It's just a couple of grains of a powder, and it's profoundly impactful for the students because they just realize how lethal, again, a lot of these synthetic opioids can be. So, again, I'm an optimist. I always try to look for the silver lining. So whilst we have this devastating impact of an opioid epidemic in America, and we do indeed have it in other countries too, we've been able to learn very quickly in the UK. There's been terrific work done by British Pain Society and NICE in terms of, again, appropriate training and management with general practitioners and chronic pain therapists and clinicians, so that we're not in the situation that we have in the USA. And what this has done is, of course, again, got the politicians, got the people's attention, and recognizing that we have to do better. It's because there is such a lack of effective therapies to treat chronic pain sufferers, so that they're just left untreated and still with their chronic pain, that's led, again, to a lot of this overdependency. So again, that's really rallied the troops, if you like, in terms of thinking about what it is we've got to do better in terms of understanding the disease of chronic pain and come up with new and novel therapies. Now, the problem and the challenge is, as I've said, is that this is a difficult one to know. Somebody else's pain, you infer, you watch. That's because pain is this subjective experience. It's your experience and it's my experience. My experience will never be your experience, and your experience will never be my experience. So by definition, it's subjective. And when something is subjective, private, yours, it can't be objectified. But what we can do is come up with better ways of trying to work out what's going on. You can't take a blood test to find out if somebody's in pain. I wish we could, but we can't. We certainly can't take a blood test to find out how much pain you're in or why you're in that pain. So again, when we look at this array of pictures of different types of situations where people are in pain, all leading into, again, the emergence of the experience by brain mechanisms, we recognize that looking, say, at these two x-rays of these two hands here, again, will bring all sorts of interesting cultural biases in terms of whether we think that this person with this very arthritic looking hand probably should be feeling pain because that feels natural to us, whereas this one probably shouldn't. And we'll feel less comfortable if somebody said, actually, this hand is excruciatingly painful. Yet we'll probably be okay if somebody said, actually, it doesn't hurt at all, because we sort of have these biases that we're brought up with in our society through our literature, through, again, medium of posters and slogans, through attitudes and biases in the medical profession, through our religious history, that prejudice us about what we feel somebody's pain should be. And this is a real problem. And this is, again, at the heart of the challenge. And so what we need are better ways that we can sort of go behind the scenes and find out what really is going on and come up with objective, concrete information that, again, can help explain why is it that this person is absolutely in pain, and you shouldn't be biased and think that they're just exaggerating or they're making it up or they're just weak. And again, this is what I hope by the end of my talk you'll be persuaded we're increasingly able to do. But what we currently do is we measure pain in three main ways. One, we use very heavily self-report. So we ask you, how much pain are you in? And we take all that suffering and all that comorbid mood alteration and job loss and sleeplessness and etc. And we say, in a 0 to 10, how much is that? I mean, it's frankly ridiculous that we do it like that. And of course, for some children and babies, they can't even speak. So we have to say, pick a picture that best matches your pain. Again, I don't need to tell you how inadequate that is as a metric. Observation of behavior, watching, seeing. But again, how people choose to express their pain is their business and will be highly variable. It'll be variable across cultures, it'll be variable across ages, and it'll be variable just because people choose to express things subjectively differently. So again, we can't make inferences just by watching behavior. Sometimes it can be helpful, but sometimes it can be not, and it can be misleading. And if we have neither of those things, because the person is anesthetized, they're in a coma, they're maybe a demented elderly person, then we have to go and use indirect physiology to again find out what maybe is going on. That will be a signal that they're in distress, that they're in suffering. But all of these are not perfect. They're all subjective in themselves, and they can profoundly lead to limitations in how we're diagnosing and how we're assessing the effectiveness of treatments. And they can even lead to real problems in, again, access to disability benefit and courts of law. So we cannot take a blood test to know if, how much, and why someone is in pain, and we need what we call pain biomarkers. We're very lucky across many other disorders of the central nervous system and diseases to have biomarkers. We have biomarkers for cancer, we have biomarkers for dementia, we have all sorts of things, but we don't really have them for pain. So there's a lot of initiatives now really born from, again, in the USA, a big National Institutes of Health funded, many hundreds of millions being poured into developing, fast tracking new treatments for chronic pain that are not addictive, don't lead to the opioid dependency. And a part of this big push is, again, recognition that we need pain biomarkers. We need measures that we can use that are scientifically robust, that are objective, that give us important information to help us, again, understand why your pain is a certain way. And then we know where to target and to try and switch it off. And a very new UK RI, Medical Research Council, has just developed this very exciting advanced pain discovery platform, and several different consortia, including one in Oxford and several around the country, are all going to be dedicated and working towards, again, developing better understanding across all the different chronic pain disorders, from mechanism, hopefully and ideally, through to translation and to impact. So pain biomarkers are essential. We need them in, for drug discovery. We need to use them in veterinary care, for all our animals. We need to use them when we've got a patient that's anesthetized or comatose, or we're trying to understand somebody who has not got verbal, or they're a baby in an ITU, or they're a demented elderly person. We need them in courts of law and insurance companies. So hopefully you can sort of get a sense of the array of different areas where it would be very, very helpful if we could understand what was going on. Well, before I get to that and how I think we can use brain imaging and give you a few examples, let's just give you the basic building blocks of the pain system. Now, all of perception, and what I mean by perception is the things that you perceive. So perception is your ability to see, your ability to smell, your ability to taste, your ability to feel touch, your ability to hear. All these basic functions that you have are all perceptions. They're all brain emergent processes. So it's only when the brain gets the signal from, say, the eyeballs or the ears, or through the tongue and the taste, or through the nose and the smell. It's only when those signals go in and the brain puts it together. Do you see? Do you hear? Do you smell? Do you taste? Do you feel touch? So perception is a brain-based process, and it's an emergent experience. Pain is a perception. There is no such thing of pain until the brain receives the signals from the tissue damaged areas, processes them, and from that emerges the experience of pain. So pain is in the brain, and pain doesn't exist until it gets up here. But all of perception, including pain, is built on what we call a 3-neuron, a neuron is just a nerve that brings signals in, system, of which one job is this thing called transduction. And that's the business end. That's the sort of bit out there that's talking to the environment. So in the transduction end, and in pain, we think of this as nociception. You've probably heard that word. So we've got these fibers under our skin that can communicate to things in the outside world that are going to hurt us, that generally, if you think about things that hurt you, they fall under 3 categories. They fall under the thermal, burning yourself on a cup of coffee, the mechanical, cutting yourself or pricking yourself, and then the chemical, could be a bee sting or a nettle sting. So those are the 3 main categories of things in the environment that are going to hurt you. And how do they do it? Well, those things communicate into the skin, and there's little nerves, the first nerve that's got these specialized ion channels, don't worry about that, and receptors that can bind those inputs from the bee or from the mechanical prod or the burning hot coffee, and they'll take those signals in and they'll start sending them in on the journey along the nerves, say from the arm into the spinal cord. So that starting point of nociception is the first starting point of this 3-neuron system upon which all of perception is built. That's called transduction. The next bit is the transmission. Send the signal in, get it in, because it's not going to be pain until the brain's got it. It's not going to be vision until the brain's got it. It's not going to be a smell until the brain's got it. So the transduction does the communication, to say the nice smell of a delicious perfume, or a nice taste of a delicious bit of chocolate. That's transduction. In pain, that's nociception. Transmission is send the signals in. So they go in on specialized cells, specialized nerves. And in the context of pain, we've learned a lot about the importance of transmission, sadly, from patients who don't feel pain, patients who are born with a congenital insensitivity to pain. So although they got the transduction, some of them have a genetic problem that the transmission doesn't work. So they receive the signals from the environment that hurt them, but they don't send them in. So that transmission bit is blocked, and it's blocked because these particular types of ion channels are not working as well. And this is a very exciting area of drug discovery, because that's taught us if you can't send the signal in, you don't feel pain. Well, let's block that transmission. Let's block it and pretend and make you a sort of almost congenital insensitivity to pain patient. So that's, again, transmission. And then the final third point is perception. And that's when the brain receives it and pain emerges. So this basic principle of transduction, transmission, perception is the basis upon which all your perceptions, vision, hearing, taste, smell, touch, works. And in the context of pain, the transduction we call nociception. The transmission, we've learned a lot from these patients who don't feel pain, and that's an area of drug discovery. And then the perception's the big one, because that's where pain actually comes about. That's where we sit. So this is where we come with all these fancy tools, which sit under the broad category of advanced neuroimaging. And we can look at the brain in so many exciting ways, guys. It's just brilliant. We can look at the brain from a structural perspective. We can look at how your brains are wired up and how good is that wiring or how weak is that wiring. We can look at the chemistry of your brain and look at neurochemicals and see whether your brain's very excitatory or quite inhibitory. We can look at conversations going on in your brains and see how well those conversations are working in really important networks that are very important for pain or very important for blocking pain. We can see how well they're working. And then we can do things like take you into the scanner and we can make your pain come about, and we can elicit whatever it is that produces your pain, or we can put pain on you by, say, burning you or electrocuting you or poking you. We have a whole little torture chamber in the Oxford lab. And then what we can see is how the brain reacts to that input. And then we can try and work out what's going on in terms of brain processes. We've got other tools that we can use. These are other techniques that you might have heard of, like electroencephalography or magnetoencephalography or EEG or MEG or positron emission tomography. And again, they give you different windows into the brain. And all together, more and more, we're trying to use many of these techniques in one patient. So we get a really complete, broad sort of physiological understanding from the wiring, from the nuts and bolts of the brain networks, from the chemistry, from the amount of receptors, how well are the electrical activity shooting around your brain. And then all together, we can piece all those wonderful different pieces of physiology so we can really understand well what's going on behind the scenes, and then relate that to, again, what you're describing in terms of your pain, what we're seeing in terms of, again, what's going on in terms of the tissue damage, and what we're able to see in terms of, again, therapies targeting different bits. So pain emerges in the brain. We knew that back in Descartes' time. So going back to Descartes, this very famous picture of this boy who put his foot in the fire. Descartes, who was a very famous philosopher, had a very simple understanding of pain. What he said was, how much damage you get equals exactly how much brain activity you get. So it's a very one-to-one linear relationship. If you burn more, you feel more pain. And generally that's true. However, it's more complex than that. And so what increasingly we know is that dependent on whether your brain is in a sad state or a happy state, if your brain is attending to the pain or distracted from the pain, if there's injury present and other mechanisms going on amplifying the circuits, if the context is different and you're expecting the pain or you're not expecting the pain, all these things will change the way the brain is sort of sitting, and that will profoundly change the way the signal coming in from the burning foot is going to be processed. We also know that there's these very powerful descending systems that can powerfully control what's allowed in after that transmission process and block it. So if you block it, less comes in, you feel less pain. So now we're in a situation where we know it's a lot more complex than what Descartes said. It's not a one-to-one mapping. There's this whole journey across the 3 neuron system where there's all these amplifiers that can turn it up and it can turn it down. So what actually ends up happening in the brain can be a far cry from what actually was going on in the periphery. This is really important. Again, going back to the judgments that people make or the inferences that people make about how much pain they think you should be in based on what they're seeing in the injury. You cannot possibly know what pain somebody's in based on looking here, because this journey is complex. It's full of these points of amplification and attenuation. And even then, when it actually gets into the brain, it can be profoundly influenced in terms of what areas it activates, and that will change the quality and the intensity of your pain experience. So hopefully already you can see just how challenging it is and why we need different tools to go beyond what somebody is saying or what we're inferring when we watch somebody, because you're not possibly going to know what somebody's private pain experience is just looking at what's going on in the injury. So it's not a simple one-to-one mapping between nociception, transduction, and perception. It's a key take-home message. And part of the early area that was identified as being important for being able to control that modulation was the this early theory, the gate theory by Patrick Wall. I'm sure you've heard about it in these classic papers back in the 70s. Where he showed that there was a capacity in the spinal cord. So after that transmission comes in, that things could be changed there. And there was an amazing amount of modulation. It wasn't just the case that the signals came in and that was just a motorway that just sent them up to the brain. Something was going on at that point when they entered that could profoundly change the processing of those signals, turn it up, turn it down, controlling again what's allowed in, like a gate. And one of the things they worked on was this gate theory, where if you sort of have a child that falls over and they hurt themselves and you rub it, not only is that distracting and that will help, but when you put an A beta in, it again, at the level of that spinal cord, it will shut a gate on then the painful scratched area of skin on the leg that has fallen over, and it will put a gate and block it so less goes up. And that's what's used in things like the TENS machine and childbirth and others. Is what you're doing is you're artificially, with the TENS machine, putting in lots of that normal touch system, just like it's like having your mother there all the time rubbing and sending those A betas in, keeping the gate shut at the spinal cord so that you could have less of those nociceptive signals going up to the brain. So very, very important landmark principle in the 70s to start the journey that what Descartes was telling us in the 1600s, it was more complex. And what Pat Wall was able to do was, through the gate theory, open our eyes to the fact that things could be changed. Things could go up and things could go down. Now we've got amazing spinal cord imaging that we're able to use, and in humans, it's not for the faint-hearted. Again, I understand that the audience is mixed between some people who have scientific backgrounds and others who are not. So I'm hoping that my slides will speak to everybody. But for those who are safe in the more sciencey background, I've always put the references in, and there's a little bit of real data just to give you the sense of what the data looks like. And spinal cord imaging, has been led by some terrific people who've come and worked in our lab, like John Brooks and Joe Kong, and then some wonderful researchers out in the in Hamburg in Germany and in the USA. So a terrific but limited still number of imaging researchers working on the spinal cord, as opposed to many of us working on the brain. And we are able, you can see in these pictures, we can actually see the human spinal cord in action, which is just fantastic. And we can see it being modulated again in different situations, which we're going to come back to when you're distracted from pain or when you have placebo analgesia, and we can actually prove that the spinal cord has been suppressed. So we're able now to sort of go back and verify a lot of these early theories and show in the human, again, what's going on in terms of, again, this capacity to modulate pain. So bringing that all a little bit together, nociception and pain are not a straightforward relationship. It's non-linear. And if we give somebody, again, a nociceptive signal, we burn them or we poke them, even if we're giving them the same burn or the same poke, and we make them depressed or anxious, or we distract them, giving your kid, the jab, or you want to get over that finishing line in a high arousal situation, even though the same amount of tissue damaging signals are coming in because of all this capacity to turn things up and down. Hopefully you can see this wide set of brain regions becoming active here, which is encoding your pain experience, all those different brain regions. And don't worry about what the brain regions are. The principles are what's important to understand here. It's just that a lot of brain activates when you feel pain. And the bits that activate basically doing the job of telling you where the pain is, what quality and intensity is the pain. It's driving you to attend to it. It's having you make a decision, what are you going to do it? It's driving action. And then, of course, you have the emotional reaction to it in terms of fear and threat. There's learning, you remember and you associate. Then there's expectations, all these different bits, complex, very multidimensional, just to a very simple pain experience, let alone a chronic pain experience, requires a lot of brain processes. So we have all these different brain regions that are available to be active, and they will be variably active, even to the same input, depending on whether, again, the brain is in an attentive state, a vigilant state, a distracted state, a happy state, a sad state. So you can just see here the graphics. For some reason, my thing is not working in terms of going up and down, but hopefully you can just see the pattern of activity there, just going up and down. And you can see, again, the blobs coming and going. And the principle of what I'm trying to say here is that even to the same input, again, these different contextual environments and mood environments in which you're experiencing that pain will change the pattern of brain activity. And if you change the pattern of brain activity, you're going to change the pain experience you have. So that's the beauty of what we're able to do now with our brain imaging tools, is go in and have a look and find out why it is that when you're distracted from pain, it doesn't hurt. Well, isn't that a useful thing to know? Because then we can bring that in as a maybe an informed therapy in cognitive behavioral therapy to really drive some of these areas of the brain that can help put the brake on and suppress it. Let's find out the bits that make it worse and are amplifiers, and then let's try and stop them. So again, we can go behind the scenes of your pain experience, find out and explain what's going on. We can explain that non-linearity between the amount of damage and then the brain experience that you're having. So some very early work we did was that we just took a bunch of students and we simulated going to the dentist and being absolutely terrified and really anxious and feeling very threatened. And even though when they were not threatened and when they were threatened, they got exactly the same stimulus. Intuitively, you probably already know the answer. When they were felt more threatened, they felt it hurt more. Why? Not because we were giving them any more tissue damage, but in the brain, anxiety, that was what they felt because they felt very threatened, acted as a little amplifier inside the brain and turned it up. And it was working through a little area around the hippocampus, actually a memory area of the brain, and this entorhinal complex. I'm going to come back to that in nocebo effects, or negative expectation of pain, after the 15-minute break. So very different. So when anxiety turns your pain up, it does it through this mechanism. When just more tissue damage turns it up, it just turns up all those lots of blobs. Completely different. All you know, or all I know, if I've just had behavior, is that you went from 6 out of 10 to 8 out of 10. But now I know that going from 6 to 8 with an anxiety amplifier is through this mechanism, and then through a tissue damaging amplifier was through that, and I can decode what's going on. In this one, we made people very sad. Now, for all these brain pictures I'm showing you, all the red and orange is the sort of increased activity. And the more red and orange it is, the more active the brain is, and generally the more pain you'll feel. So what you can see in this case is when subjects were experiencing, again, the same amount of nociceptive tissue damage, but they were in a neutral mood, as opposed to a sad, depressed mood, profoundly increased amount of activity, as if the state of depression was unleashing the ability for the brain to process those signals. Areas that normally would inhibit them are now not working. And so the floodgates are open and more brain is allowed to be active. So it hurts more and it feels more painful. And if you subtract all these brain blobs from these, you're left with all this extra amount of activity, not driven by burning the person anymore, but driven by being sad. And so again, it helps drive home the importance, the absolute importance of these comorbid issues that patients suffer from, because they are physiological amplifiers, but they're working inside the brain. And then the brain imaging has been able to teach us that now. What do we do about that? Well, then we'd have to treat the depression and anxiety as importantly as we're treating where the tissue damage is, because the sad truth about chronic pain is that that 3 neuron system, transduction, transmission, and perception, in the chronic pain state, generally all 3 of those elements go wrong. So we've got to fix it everywhere in order to really do a good job. And again, this is just to emphasize that when pain emerges by brain mechanisms, there are brain mechanisms in there, I'm afraid, that can make it worse, but they can also make it better. And we are going to cover those, I promise you. So bit of Blue sky stuff, just for a few slides, is just sort of going back to sort of which bits of the brain actually encode, ouch, that hurts. You know, where's the basic neuroscience understanding of, if you were going to build a brain to feel pain, what would you put in it? So we can do this by using what we call brain reading, approaches. You've probably heard a lot about artificial intelligence and machine learning and the fact robots are going to take over the world. Well, what we can do with brain mapping techniques now is we can use brain reading approaches to actually do some really cool things. So normally in our imaging experiments, we'll take a person and we'll burn them, or we'll poke them, and we look at the brain response to that. And then we don't burn them or poke them, we do something just less, and we look at the brain response to that. And we subtract all the brain activity here from all the brain activity here, and we get a pattern that says these are the bits that are important in the difference between pain and no pain. And then what we can do is do that lots and lots of times in lots of different situations: squeezing painful joints, touching areas of burning skin, constant ongoing throbbing pain, any type of pain you want. And we can train up computers to recognize brain patterns that say that was when they said it hurt, that was when they said it didn't, that was when it was really high, that was when it was really low. And then what we can do is throw in something that we don't have an ability to know what's going on. I always joke and say, okay, well, we'll put a Neymar here. He's always a bit of a faker when it comes to the football, wanting to get the free kick. I mean, is he really in pain? But in more seriousness, situations where we've maybe got babies in neonatal units, or we've got people anesthetized, where we'd want to know, is their brain pattern telling me that they're in this bin or this bin? Because I can't ask them, and I've got no movement here to infer. So what you can do is put then their brain pattern into the computer, and it will then do what we call a machine learning, and go back and say, with a probability, 90% we think it's pain, or 90% we think it's not. And this is very early days. This is very crude at this point. There's lots of issues and problems, with it that we can go into in questioning. But, you can see how this could potentially be very useful going forward, particularly as we build more confidence, as we build up the databases and we have more and more data going in to help refine this machine learning, how we could then use this in tricky situations where we really want to know if somebody's in pain and we just don't have the luxury to ask them. So that's, again, a big area, but also it's an area that gives us an opportunity to learn out of that complex pattern of brain blobs, which bits are really essential if you were going to really just have a pain just above threshold and a brain map just below threshold. Is there something we could learn about what the essential bits are that encode pain? Because that's still the journey we're on. Again, those would be targets we might want to go for. So a lot of machine learning is being applied to, again, understand these situations where you've got a sleeping baby or you've got an anesthetized person, but also they can tell us a lot as well about what the key bits are of the brain. So let's just think about sleeping babies. I mean, this experiment, which I was lucky to be involved with a very dear colleague, Rebecca Slater, who's doing wonderful work in brain imaging of challenging, again, 1-2-day-old babies, very premature babies, and then watching child development. Until fairly recently, staggeringly, it was thought that babies don't feel pain. They just don't feel it in the same way as us, because their brains are not very well developed. All the folds and things that you normally see in a human brain are not very folded in a baby's brain. And going back to a German anatomist in the 1800s said, well, because they don't really have a folded brain with all the gray matter ready to go, they can't possibly be processing complex things like pain. So they probably don't feel it, don't worry about them. And staggeringly, that view persisted for a long time. So obviously, it's been overturned in latter decades, but only in latter decades. And so what's been very interesting about, again, bringing in some of these brain imaging tools is to prove unarguably that even a baby that is 24 hours old, this is one, put in a scanner, put big ear defenders on because it's very loud in our scanners, and poke their little feet with a little needle, which is the same as a heel lance that they would have naturally to take a bit of blood. Parents, absolutely wonderful in supporting these studies in the local hospital here. And this study was done, one, to just sort of prove, no, they do activate, but also it gave us the opportunity to say, well, if you're a 1-day-old baby and you're asleep inside the scanner, it probably will give us the basic set of simple brain regions you need to activate to feel pain. Because at this point, the baby has not learned to associate and anticipate pain and have fear of it, learn from it. So all the extra bits that we bring on as we become adults and we have lots of pain experiences, they haven't got that. So it would be a very stripped down basic set of brain regions. It would be really helpful for us from a basic science perspective. And do you know what the answer was to that experiment? Basically, 24 hours later from being born, fast asleep inside the scanner, their brains don't look any different basically to human adult brains. It was the same. It was all good to go. I mean, just speaks to how important it is to experience pain and the fact the brain networks are ready to go, ready to be active. There's a few little differences, but largely, very, very similar to the adult brain, which I think is just remarkable. So again, that's teaching us something important, that these areas of the brain that encode where, what, how much, and attend, I don't like it, I better learn from it, ready to go on day one. And then what we can do, if you just look here in this sort of more complex anesthesia work that we do, where we just take people into deeper and deeper states of unawareness, as you would in operation, is we can be bombarding them with, again, laser stimuli that are painful like pinpricks, and we can watch how the brain just switches off and strips down. And then that can teach us again a little bit more about which seems to be the bits that are taken out and how does that relate to how the person's, again, reaction to things that are painful occur. So again, these different types of ways, including the brain reading, are new ways in the basic science, discovery science, that we're trying to use to give us that core basic discovery about, if you're going to build a brain to feel pain, what would you put in it? Which bits would you put in? I think we've got some good ideas now. And one of the other things I think that brain mapping has also helped us do is to disambiguate that when you are experiencing somebody else's pain through empathy, through social pain, through emotional pain, I mean, it is a fact that in our society, our language is riddled with words related to pain when we describe things of great suffering. It's the go-to word. And, we have, this ability, because it's what makes us social animals, is that ability to have empathy, to feel another's pain, so that we can put ourselves in their shoes and be good citizens. And as a society and as animals, that helps us go forward collectively. So this is why we have this ability, these wonderful quotes in terms of, again, how we have to imagine somebody else's suffering, but it's an important skill we have. So some of the early questions were, well, if we are experiencing non-nociceptive and we're experiencing emotional pain, how different is that to nociceptive pain? So there were several experiments done from, again, empathy studies, watching the brain react as you're watching this type of thing, which just looks terribly painful, watching your partner being burnt, and looking at your brain as you were watching your partner being burnt, and then looking at your brain as you were being burnt and saying, is it different? Is it similar? And in the early days of those studies being done, studies looking at social exclusion and social pain, not being, setting up scenarios like, everybody's lining up to be picked for the netball team and you're the last one standing and you feel excluded. It's all happened. And again, you'd feel that as you felt hurt, being dumped by your partner. You feel hurt. You use the word hurt and pain, grief. So imaging people's brains, you could see that a lot of the brain areas were very similar to that pattern I showed you earlier, that bilateral, those big blobs everywhere around the brain. But the brain reading, the machine learning analysis has given us a fine-grained look in to say, actually, there's some really very important differences in it, which largely relate to, again, how the signals are coming up and going to the sensory discriminatory. Where is it? How intense? But for a lot of the more emotional affective areas, it's a very clear overlap. And that's important. And that's why we probably use those words of social pain and emotional pain. Hypnosis studies have also been done and hypnotize people to feel pain to a non-nociceptive input and vice versa. There's some very recent work just published showing this capacity for, again, animals to pass over, again, empathic pain to each other in mice and pain relief in each other. So this is cross species. This isn't just a preserve of us. This is animal behavior. And it's important because it's what makes a good society and it's what protects us. But it's important also to know, not because, and this is really important, not because you're making a judgment call that nociceptive driven or physical pain, as it's sometimes referred to, is more important than emotional or non-nociceptive driven pain. Because if, again, pain is something that emerges by the brain, and I'm agnostic as to whether that's emerged because there's been a nociceptive input or whether it's emerged through brain mechanisms. At that point, it doesn't really matter. It matters to know which one, because then you treat it in the periphery and stop it coming in, as opposed to if it's not being driven by there, it's driven by the brain. But we have to move better forward in a society that we don't treat emotional pain as a second-class citizen to physical pain, or nociceptive versus non-nociceptive. And I'm really very passionate about that. And I think brain imaging and the brain reading approaches have been really instructive in giving us that capacity to know the difference, but to know the similarities, and then to contribute to the discussion that if one is in courts of law, for example, and it's an emotional pain, not a non-nociceptive, well, that's pain. Pain is pain is pain. It's a brain pain thing. It's important to know which one is driving it, but we shouldn't be making judgment calls about which one should get benefit and which one shouldn't, in my opinion. And again, we can discuss that. And this is an evolving story. And again, this is where brain imaging, I think, is contributing in very interesting ways. Okay, switching to chronic pain and the core mechanisms. As you know, many different types of conditions, there are specific symptoms and there are shared symptoms. And the big one that many of you are suffering is the constant ongoing pain. And in terms of what we strip down to try and understand using our tools in terms of what holds people in chronic pain is really that, understanding of why are those fibers, those fibers that are bringing that signal in from transduction transmission, when they're damaged, why do they switch on and just keep sending the signals in? Let's just stop that. And let's have a brain imaging tool that can look at that constant firing, because that can then tell us where those signals are going in and we can hopefully switch them off. The central nervous system, which is the spinal cord and the brain, if they are receiving constant signals coming in from your damaged nerves, I'm afraid they don't just sit there and take it all day, being battered like the waves on the wall of the seawall. They respond and they adapt, and they adapt in maladaptive ways, and they adapt in amplification ways. So then they set off these amplifications. So they actually make it worse, I'm afraid. If things are left sort of not treated and just ongoing, then we're very concerned about, again, rewiring and maladaptive plasticity. And that's, again, an area that's still being looked at. And then the really sort of late area that we're interested in is, who is that 1 in 5? Why is it 4 out of 5 don't end up and stay in a persistent state, but 1 in 5 do? What is it about, again, issues around vulnerability and resilience in terms of, again, what might have been the patient's journey to the point that they got injured that might, again, have developed a resilience or a vulnerability in some of these core networks. And again, that's a very new area, but a very exciting area for research. And again, I'll touch on that at the very end after the break. So in terms of ongoing pain, we've been working to develop a new imaging tool that's enabled us to give us some signals there, so that we're able to find out what's the sort of readout, if you like, in the brain of just a constant, ongoing, slowly varying pain. And we've identified a couple of brain regions that seem to be really important. And then we've been working with our neurosurgeons to go in and show that in patients, we can verify that, say, these key bits of the brain that we've shown in this basic science study indeed were active there in patients. And then we've gone in with a surgical implant. And again, it's a new area, a new target born from, again, the discovery science to try and modulate that. And we're still analyzing this data and collecting the pain diary data, but I wanted to share this with you. And that we're getting, really effective interventional therapy in, again, some of these new targets. So this is a nice example of, again, discovering a new discovery in terms of what's encoding that constant barrage of inputs, a new technique to allow us to discover that for the first time, and then translating that out into the clinic with our wonderful neurosurgeons, Alex Green and others, to, again, provide a new target for deep brain stimulation. This area of the brain, I mean, it's not to say it's the only area of the brain, but it's quite a fundamental core area of the brain that passes on the signals to lots of other areas. So if you bring it down, you'll be bringing down quite a lot. It has been shown in many years ago from wonderful colleagues at Michigan in the USA who work largely in fibromyalgia, that this area of the brain, and again, don't worry about what these brain areas are, it's the principles of the matter. If you measure chemical information in it, it turns out that in fibromyalgia patients, it's very excited. There's a lot of excitatory chemicals there, this thing called glutamate. They've got more glutamate there than controls do. And the more glutamate they've got there, the less you need to squeeze to elicit a sort of 8 out of 10 pain to the squeeze. So if they've got lots of this excitatory neurochemical, you only need to squeeze a little bit for it to be out of 10. If they've got less of that excitatory, which means that the region's not going to be as active as quickly, you got to press a bit more to drive that region up to make it encode pain. And again, our studies were showing how important it is for encoding constant and ongoing pain. And moreover, this signal in this region and this chemical, if you can calm it down, so those patients who were very well treated with, in this case, pregabalin, it turned out that the people who had more of this excitatory level, the more effective the pregabalin was. And what did it do? Well, it brought down the amount of that excitatory. It just calmed it down. And the more raised it was before the treatment, the more predictive it was of good outcome. Really, really exciting. And again, it's sort of pretty basic, simple principles, but so beautiful that one's able to show that. And again, this is because we've got these different tools to look at, again, different windows of the brain here. And again, looking at an overexcited brain that's, again, driving regions that, again, these other studies have shown very important for encoding, again, constant pain and the intensity of pain. The amplification, remember I said that if that signal is constantly going in, the spinal cord and the brain doesn't just sit there taking it, it changes. And what it sadly does is it sets off this thing called central sensitization. And a very important added dimension to that, which is this thing called the descending pain modulatory system, is this very old brain system in the brainstem, which has this lovely wiring to, again, old brain regions, that your basic fight and flight regions and fear regions, like the amygdala, your sort of stress axis systems like the hypothalamus, beautifully wired up to even more higher order, decision making, mood areas, expectation, attention areas, all can be wired up down to the oldest part of the brain, the brainstem, to drive this exquisite system, the periconductal gray and the rostral ventral medial medulla, which talk all the way down to the spinal cord. You might have heard of a thing called the dorsal horn, and that's the area of the spinal cord that the tissue damaging signals are coming into. And what it does is it communicates down to this in an anti-nociceptive way. That's the good cop, it inhibits it, or a pro-nociceptive way, that's the bad cop, it facilitates it, turns it up. Now, this first level of amplification of central sensitization is already occurring at the dorsal horn of the spinal cord to that constant input of signals. This thing has set off this amplification thing called central sensitization. On top of it, you've got the bad cop going doolally and now making it even worse and it keeping it going. So you've got these layers of amplification that again are amplifying those nociceptive inputs. So more goes in and it hurts more. And this is now, we know, this imbalance, too much bad cop, not enough good cop, is what is very important in maintaining people in these persistent chronic pain states. And again, this is becoming something that we understand better and better, both from a lot of the work that we've done, but also a lot of preclinical animal research, again, working on this system and showing the importance of this system in, again, this layer of amplification. So things like allodynia in normal clothes touching you, hyperalgesia, I think they're more painful. All of these things can be explained, and again, with more detail, but I won't go into it, due to these, again, amplification phenomenon. So we've shown in a range of different conditions, people going in for new hips, people going in for new knees, having surgery. We've stratified the patients ahead of time and said, let's just find out how much before you have your new hip and before you have your new knee, come into the lab and we'll put you in the scanner and we'll just see what's the balance between your good cop and bad cop. And we'll do some other measures on you just to see whether you've got, not just nociceptive inflammatory problem, but do you have a bit of a neuropathic-like element maybe to your arthritic knee or hip? And what we were able to show is in about a third of the patients, indeed, before surgery, we could show they had a lot of bad cop and they had neuropathic-like elements. And we've done this both in hips and we've done this in knees. And then what we've been able to show is that a year later, if you ask them whether they're in moderate to severe pain with their new knee, you've got a group that say, yes, actually, I'm not any better than I hoped I'd be. I'm still in moderate to severe pain. And do you know what? They were the guys that had the bad cop ahead of the surgery. So again, this is a nice example of discovering and showing a mechanism that's very important that we could see with our brain imaging tools, and then translating it out into the clinic again and working with our surgical colleagues to again see if this is a useful predictor of outcome. Of course, what are we going to do now? Are we going to recommend not to have that? Well, again, these are where the studies need to lead and need to go in terms of how do we use this information now? Is it a case that maybe it's gone too long for that patient and they were left too long before they got the new knee and hip and they went more central and it was allowed to get to a place where there was more bad cop? Was it that even before they got an arthritis, they had too much bad cop? Is that part of the vulnerability and resilience issue I've been mentioning? And again, that's more my hypothesis, a little bit more what we're pursuing in some other studies, is again whether this is already a little bit wrong, and that's what's predicting again that element of the nature of their pain, and then again being predictive of not so effective afterwards with the therapy. I don't know if any of you have diabetes and diabetic painful neuropathy, but we've done a study with our colleague here, Dave Bennett, where we looked at patients with neuropathy, but one group had pain with it and one group did not have pain with it. And very difficult to see anything in the periphery that could explain that. When we looked at the brains, and we looked at both what the brain was up to in terms of just the range of pain that the painful patients were describing, and then what the range of pain was when the patients very, very gratefully allowed us to put hot water bottles on their legs and really drive hyperalgesia. Again, they gave us their behavioral description of that. We measured the imaging response of those hot water bottles. And again, what we were able to show both for their own just background pain and then their hyperalgesic pain to the hot water bottles was, surprise, surprise, the group that had more ratcheting up of that bad cop, that explained the range of clinical pain. It explained more communication to these other brain areas that are encoding pain. And it explained how much heat hyperalgesia they were going to have both behaviorally and with the brain imaging metric. So again, it's not to say this is the sole explanation of pain in diabetic painful neuropathy, but it certainly is contributing in a very significant way. And that's important because it means we're now looking in different places than we maybe initially would think in terms of treating the pain out of the legs. We've got to think about what's going on in the central nervous system. Where can we turn things down there? And again, a lot of these systems in the brainstem are can be reset with, again, different drugs, and I think also different interventional therapies, non-invasive brain stimulation, and different cognitive interventions that can access some of these systems, which is again providing alternative ways to reset things. This is a very complex side, and the only point I want you to learn here is that this gives us this signal that we can measure here that we've shown is important for stratifying, and we've shown is important in a range of different chronic pain conditions. We can now use as a biomarker with industry for drug discovery. They have a really terrible time trying to get drugs out to know which drug is going to work and which drug is not going to work. And so to earlier on get rid of the ones that are not going to work and to pick the winner. So we've done a lot of work with industry over the years, and we're in lots of consortium at the moment. I just want to give you a lot of hope and positivity that despite the fact it's been a bleak period and a graveyard really for analgesic drug discovery, things are looking really good. There's new ways that we're doing our clinical trials, and we're bringing in more than just 0 to 10 on our rating scales to assess whether drugs are working. We're looking in the brain and saying, are you actually doing anything in there? Are you actually suppressing activity in bits that we know are important? So we're using this as a biomarker, and that's really exciting. And that's, again, ongoing work at the moment. Well, the good cop is there. And so again, this is something that we can just use in everyday distraction analgesia. I call it free analgesia. So this idea that we've got the capacity to drive that good cop, and again, that can be driven by this system in the brain that is, again, can be driven by very, very frontal areas right here. And these are areas that can be, again, in different situations of attention and expectation, or reframing, rethinking what your pain is. You're driving in a domino fashion that network, and you're boosting the good cop. You're flooding then the brain with endogenous opioids, your endorphins. You're blocking the signals here at this entry point where those tissue damaging signals are coming in. You'll just say, no way, you're not coming up. So they don't come up, you don't feel pain. So that's why when you are high arousal, distracted, you don't feel pain. And the key question that was first asked was, is that system, the good cop, the system that placebo analgesia uses when it has its effect? And basically the short answer is, indeed, that is what it uses. I think for time, I'll skip over that. This slide was just to show you that system you can drive with this little brain device. And this is what we call it's called non-invasive brain stimulation, which means you don't have to have surgery. There's no probes inside your noggin. You've just got this patch here attached to a 9-volt battery pack, and you can drive and tickle then the start of the brain here, because these regions can be accessed through that patch, and it can cascade and drive that descending system. So we were able to show that indeed we can drive that system, and we can show that we've got suppression of, again, that region that I showed you was encoding ongoing pain, we can show tonic suppression of it with effective brain stimulation. And what we were able to also use is one of our other structural imaging methods to look at how good is your brain wiring between the bit that we're stimulating and the bit it needs to pass down the signal. And do you know what? The more well wired up you were, the people up here, the more effective that therapy was, the more analgesias you got. And the people who had less good wiring, we've got these measures to measure that down here, had less good analgesia. So this was a lovely study to show one of the most basic principles of all of biology, that structure and behavior and function all sort of go together. And even just subtle differences in people's fundamental wiring explained how effective or not effective this therapy was. And it makes total sense, because if that motorway is not working as well, you can't send the signal down, so you don't get as much analgesia. And for some of you, you just naturally have good wiring, and it worked even better. So again, we wouldn't have known that. We would have just seen, well, people had good or bad outcomes. Why? Because everybody's brains are a bit different. And that difference, we can say, right, you go for this because this is going to work for you. You guys have to go for something else because you haven't got that as well. And that's how we can start to use these different windows into the brain to help us better decide earlier which intervention is going to work for you. And that's the key, isn't it? So placebo is a really massive thing. And I think what I'm going to do, short of just sort of, again, highlighting its historical thing, which is really interesting, and I'll probably go into that in the questions, because there's a lot of confusion about placebo. Even the very word evokes deception and fakery, because it's the word for these chants that monks would sing at your funeral, but they were paid to turn up to your funeral, so they were fakers. And that's why it has this connotation of fake and deception. That's a real shame, because actually until recently, which is when this all went confused, we really understood that the placebo effect and how one set up expectations were powerful, and they really influenced outcomes. And what the science has taught us is there's nothing special about placebo analgesia, and it's certainly not fake. It basically just grabs the good cop. And again, that's been shown with beautiful brain imaging showing this system. It's been shown with spinal cord imaging that you can suppress the activity here in placebo analgesia, and this is it, not in placebo. So again, it's unarguable that this thing is powerful, it's physiological, it's neurochemical, and we know it really well. Now, the trick is, how are we going to use it in modern medicine? Because you can't lie to the patient. But what you're doing in a placebo manipulation is you're setting up expectations, just like Harry Potter did with Ronald. With this, you're just setting up expectations. And that's really a very core aspect of a placebo manipulation. You set up those expectations, you for free access this amazing system. So how do we set up expectations? Well, in the clinic, that's what's happening. So at this point, I've only got about 10 more slides, guys. But at this point, I think we'll take a break for, say, 15 minutes, and then we'll come back and I'll finish off with expectation in the therapeutic setting and this concept of you getting the pain you expect, and just where final slides, sort of where we're going now, and then open for questions. So is that okay, Nikki, if we just take is this a good time to take a 15-minute break?
Yes, it's
ready to go. Great. Okay, well, welcome back, everybody. I hope you're all refreshed and ready for a little bit of discussion around expectations. So this issue is really, really interesting and very, very important, I think, and was understood, right back in the mists of time from Hippocrates and Galen, understood that they just didn't have the science to sort of explain it, but they understood that that social contract that comes between the patient and the physician, or the patient and a carer, is important, and it changes outcomes. What we've been able to do more lately with the scientific approaches that we've got is to actually sort of understand at a physiological level what is actually going on in that important, patient physician or patient important partner interaction. So when you as patients go to your healthcare professional, you'll go with obviously knowledge of your pain, experience of what's worked for you or what hasn't worked for you. You'll go with a lot of knowledge about things that you've read, and then expectations about what you hope the next meeting with them will bring, or the new intervention they might be offering will do for you. So that is a lot going on, and that isn't sort of just sitting there. That is actually driving networks in your brain. That is changing, again, the substrate of your brain from a neurochemical perspective. So, that's the brain in a sort of variable expectancy state, which is meaningful in terms of the physiology of what's going on. And then you have the chat with the physician, and they'll set up, they'll sort of help you work through the expectations that maybe are wrong, because maybe just misunderstood things, confirm and validate the ones that you've got, and then they'll, through that consult, set up new expectations. What we know is that patients don't remember too much of what's gone on in that sort of pressured moment of that brief window, and they'll generally only remember, a small percentage of what the conversation was about. And the sort of downside of this is that it's not a consistent necessarily interaction, and different patients will walk out the room with different expectancies set up, and that will be also dispositional, in terms of whether they're more of a glass half full or glass half empty type person. They're going to have listened to more the negative things that were said versus the positive. And again, how much is remembered, all these factors will come in, that basically they'll walk out the room and all rules are off in terms of what the physician thinks the person has understood, or what their expectancies are when they've left. And that change in expectancy then is combined with the treatment. So let's assume it's a drug, but it could be another intervention, it could be a course of CBT. And it's the combination of these two things that actually will dictate the outcome. And this turns out to be very important. So again, our focus in a sort of very medical model world is that it's all about the drug, it's all about the intervention. But what the suspicion has been is that, going back from age-old time, is that actually the expectancies are probably as important and interact in interesting ways with the intervention. So let's try and find out what's going on in that context. And that's sort of what led us to do this experiment. It's quite old now, but it stood the test of time. And it's an experiment that explores not doing another placebo experiment, because, we've done those, but to say, well, actually in the real life setting, patients are on treatments, but the expectancies are variable. And that's actually what you're doing in the placebo manipulations. You're changing expectancies, and that's driving that good cop, or it's not driving the good cop. So why don't we just see how powerful expectancies are? So what we did is we took subjects, we put them in the scanner, we're looking at their brains, and then we give them a thermal painful stimulus. And they have an IV line attached to an infusion pump that's giving them remifentanil, which is a fast-acting new opioid, good analgesic. Now, what we did is we just changed people's expectancies about when they were getting the drug, or when they thought they were getting it, or when they thought that it might be stopped. So what we did is this: we brought them in, we were burning them away and looking at their pain ratings and their brain response to that thermal stimulus. And then we started the infusion of the drug, but we just didn't tell them we'd started. And then we did the same thermal pain stimulus, looked at the brains, looked at the behavior. Then we sent the anaesthetist into the room to check their heart rate and blood pressure, and to tell them, we're going to start the infusion now. We're going to give you that wonderful analgesic, setting up positive expectancies. Now, the patients have already been on the drug for 20 minutes. They just didn't know it. So at this point, we're driving positive expectancies. After another 20 minutes of giving them the thermal painful stimulus, looking at the brains and looking at the behavioral response, we then went into the room and pretended that we were going to stop the infusion of the drug, and that they were going to go back to no drug, that baseline rating. So we're setting up negative expectancy. Fancy word for that is nocebo. And what we wanted to know was whether it made any difference whatsoever. At the end of the day, you got this fantastic drug that we know works really well. Is that the only thing that matters, or did expectancy matter? And the results were absolutely remarkable. So these are the pain ratings when they were at baseline. So this is out of 100, 65 is moderate to strong pain. When we start the infusion of, again, one of our most powerful acute pain medications, I am pleased to report it works. So there's about a 10-point reduction on a 100-point scale. When we then just for free couple that with positive expectancy, we actually got even more analgesia than the drug itself. So this is about 10, this is about 20 points. So that, again, is sort of a placebo, but placebo on drug. When we then pretended that we'd stop the infusion and we mounted anxiety and negative expectancy, we actually thought in the experiment we were just going to go back to this, override that positive gain, that free analgesia. But do you know what happened? They went right back to baseline pain ratings. So negative expectancy, the anxiety of, oh my God, the drug is stopping now, I'm going to have all that pain again, was enough to actually override the drugs working profoundly. And so this is an important, because this is, again, a very important component of, again, how negative expectancies, one drug failing, then another drug failing, another treatment, can really be playing havoc with, again, outcomes. And we need to understand the outcome. This is a subjective outcome. This is a report. But if we looked at the brains, was it that even though their report went back to baseline, were their brains telling us there was still suppression of activity? Because the drug's still in there. So again, it shows you the sometimes lack of usefulness of the subjective report, because we can make it do things in weird and wonderful ways by playing tricks, if you like, on expectancy. Also, if you were doing, again, drug discovery and you were trying to develop that drug and you were GlaxoSmithKline, you'd look at these ratings and you'd say, well, this drug is rubbish. It's not producing any difference. But again, you'd be misleading. This is a great drug, but you'd be fooled and you'd throw the drug out in the script because you haven't understood that expectancies were real, were playing out through physiology, but they're playing out on top of, again, the drug working. So again, the brain imaging was able to show us, we can see suppression. So we, again, I'm not showing the slides, but we can show that we've got suppression of the brain, more suppression of the brain. And then even though the ratings went back to baseline, the brain activity didn't go back because, of course, you've got a head full of opioids suppressing it. So that was really nice to show that mismatch, if you like, between, again, what the brain imaging is never lying and not telling us something that's wrong. It's telling us that the drug is in there, that it's working. So don't throw the drug out. It is a good drug, but the behavior, again, you've got to be careful because expectancy is driving a different mechanism. And that's now adding on top of what the drug is doing. And you wouldn't know that just from measuring ratings. It just shows the inadequacy of how we're rating things. And we're not getting enough information from what's going on. And then what we're able to do is look at the mechanism. Well, what's driving that positive gain? Well, it's that lovely front of the brain driving the good cop, was what was driving all that positive free analgesia when they just were told that they're going to get the drug, and we drove positive expectancy. And then our little friend, which was our anxiety amplifier right at the start of my talk, this was the region that became more and more active when we told them we were stopping the infusion, but we weren't, but they were anxious about that. And the more anxious they got, the more that drove, and the more that drove the ratings back to baseline levels. So again, coming nicely together with work that we've done many, many years ago. And this again being a nocebo effect. So in terms of, again, this expectancy, clearly you can see it's a very profound effect. And again, Ulrika, who worked with me on this, she's now following this up back in Germany with lovely work building on this. Rather than like in an acute experiment like that, she's doing it week after week, changing people's expectancies about drugs that they're on and showing how important it is as part of the equation to drive outcomes. But importantly, how do you, and I'm using the word manipulate, not in a bad way, in a positive way, how can we harness this very powerful physiological expectation thing for the benefit and get an extra bit of analgesia for free? And again, that's work that's ongoing and really exciting to think about. But going even more into sort of, again, expectancies and belief states, I just want to, again, just pause and just tell you a little bit about how the brain works, because this sets the scene for, again, thinking at an even higher level about how you expect things and how your expectancies shape what your experience actually is. So the way that I taught you about the brain and perception was a bottom-up view: transduction, transmission, perception. Things come up and in, the brain receives them, and you see, you hear, you taste, you smell, you feel pain. It's a very bottom-up approach. And that largely is how we teach how the brain works in the three-order neuron system, and that's true. But this model assumes the brain's just sitting there, blank, silent, waiting for signals to come in, and nothing's going on prior to that. Well, that's a very inefficient way to build a brain. So, you don't want to just be sitting here completely blank, and every time you do anything or see anything, it's sort of got to start from square one, processing it all, and then seeing, whatever it is you're seeing, or hearing whatever it is you're hearing. So the brain, we think, doesn't work like that. We now think of the brain as being what we call a Bayesian brain, which is a fancy word. Don't worry about that. I can go into questions. That is not a simple just receiving organ. It's already got networks active, even at rest, that are your sort of expectancy networks, if you like, as to what you think that thing might smell like or see like. Often, I don't know whether you've ever sort of tasted something you know, like a strawberry that tastes like a banana, it really throws you because you're looking at the strawberry, it's red, you're expecting it to be a strawberry, and then it tastes like a banana if you've been tricked and things like that. And even if you haven't, you can imagine how you'd be sort of questioning. And or somebody says to you, I've done this blinded with students, you know, hidden the glasses of wine and said one's white and one's red, keep them all at the same temperature. And if I tell them they're white and red, they'll think they are. It's extraordinary. You can just, you know, change people's expectancies. So the brain is efficiently already working, thinking, what's that going to feel like? Now, the analogy I'm going to use is, can you see my water glass here? I hope you can. Yeah. So if I wasn't touching it and I had a thing, I'd be able to, but you know, I'm going to touch it now with my right hand. So this is a glass mug with water in. I already now, before I touch it with my right hand, have a brain network going on in my head, what we call a prior probability network going off, saying to me what already I think that's going to feel like when I touch it. That's happening now, even before I touch it. Now I touch it and all those bottom-up signals are coming in. But it's the combination of what I've already thought that's going to feel like and the actual feel. When they come together, that's what I feel. It's the combination of the two. So the priors, or the expectancy networks, really can shape what ultimately your perception is, irrespective of what's coming in. And we're starting to really appreciate how important these expectancy networks are. They're called priors, actually, in terms of driving all of perception. So to sort of prove this point to you, this is what your brain is actually doing. If I took you into my scanner and I lay you down, I looked at your brain in action whilst you were doing absolutely nothing. So lights off, sounds off, you're not touching anything, you're not moving. I'm having you just think as best you can, just nothing, just go into like a, you know, state of mindfulness, if you like, just nothing going on. Well, not mindfulness, that would be actually be focusing on one thing. So just try and relax and go into nothing. So there's no bottom-up input. There's nothing. You're not deciding. You're not thinking about what you're going to have for dinner later on or anything like that. You might think your brain is just boringly sitting there, just chugging along. Well, this is actually what your brain will look like. You can see it's very busy. And what we've done is we've color-coded, say in blue, all the bits that activate as a network in blue that are talking to each other. And there's another set of regions in green, and those are highlighted in green because they're the ones talking to each other. They're not necessarily talking to the blue. And then the pink are the ones that are the different brain regions talking together in pink. And when you look at what those pink, blue, green, different networks are, you say, oh, that's the network that activates when you attend, or when you move, or when you feel something, or when you remember something. So they're all your basic networks that subserve all the things that make you be able to do what you do. And even when you're not doing the thing that makes you do what you do, even at rest, they're on, and we can measure them on. So you can look at those as a way of thinking about, well, if you can do that at rest, just before I'm going to attend to something, that's on. Just before I touch something, I've already got a network telling me what the sensory network's on. And if that's really dominant, that's going to override maybe what even comes in and decide what my perception is. So that's a long-winded way of saying that these priors can bias perceptions. It's like a loaded dice. If the priors keep being built up so that every time, say, you get up, it hurts, you're going to start very quickly expecting it always to hurt every time you get up. You're going to be training your brain to expect that, and you're going to be loading the dice such that it'll always be, yes, that hurt. Yes, that hurt. So even before you get up, your priors will be going along like mad, saying, oh, that's going to hurt now because I'm going to get up to make the cup of tea. Well, maybe in that particular instance, there wasn't a trigger, but you've already set the network off and you're going to bias it to be such. And again, this sort of, you know, expecting that a nail's gone through your foot when it hasn't, that's going to hurt like hell because you expect it to. You're going to set off those brain networks. And what I've shown you here is the proof that you can set off brain networks without any input coming into the brain. They can do it all on their own. It's phenomenal, isn't it? Absolutely phenomenal. So what we're realizing is we've got to start working with priors and expectancies, particularly in cases where the pain's been left, you know, unmanaged, untreated for many, many years. This becomes now very important as an additional way to target things, in addition to obviously trying to calm everything down coming in. And I wanted just to share that latest bit of brain science with you, because it's really changing the way we're looking at many different disorders of the central nervous system, because it's teaching us new ways to think about, again, treatments and what we need to be focusing on. Well, extrapolating the expectancy into a translational level, I've shown you one with the drugs. This is some work I did with my surgical colleagues who I did a lot of the knees and hips with. This is a fascinating study where my colleagues who, particularly one of my main collaborators, wonderful, brilliant orthopedic surgeon, Andy Carr, who ran the department here. He's a very, very major shoulder surgeon and works on this particular painful condition of the shoulder where you take off this bone spur on the acromion joint. And after all the sort of work we've been doing, it sort of started to question, well, is it actually mechanistically doing anything, or is this a placebo effect? Because coming into hospital, staying overnight, several nights, having an operation, this is big expectancy manipulation. Yeah. And unlike drug discovery, which is only and can only drugs be approved if they've been tested against a control arm, surgeries don't have to be tested against a control arm. They just get done. Now, if the surgery's target is for an outcome that's subjective, so the main purpose of the surgery is to treat a pain, as opposed to fixing a blood vessel. You don't need a control thing to fix a leaking blood vessel or take a tumor out. But if your surgery is targeted for something subjective, you have got to have a placebo arm in order to be sure that what you're exposing the patient to in terms of the anesthetic and the operation and the visit in the hospital actually has legs in terms of proof that this is doing what you think it does from a pain perspective. Or is it that you're setting up these expectations and it's a placebo analgesia mechanism? So they did this study. We're still analyzing the imaging data, but the behavioral data is published already. This caused an absolute maelstrom when it came out, you can imagine. People went bananas about it, particularly in countries where this is a very major private health operation. So there's a lot of monetary considerations again in this, but they were just phenomenal investigators. They stuck to the guns. There's been now confirmatory proof of this subsequently, and as I say, we have the imaging data for a subset of these patients as well that we're analyzing. But the behavioral data was such, on this scale, the higher the number, the less pain you're in. And this is at baseline before the surgery, this is at 6 months, and this is at 12 months. One group of subjects had no treatment, it was just natural course, and even they improved actually over a year. The red and the blue are the two patient cohorts who came into the hospital. They had the anesthetic. One group had the decompression, and the other group had nothing. So it's a sham surgery, and there's absolutely no significant difference between the 6 months or a year. So it is through placebo mechanisms. If that is what's needed to drive it, we can have a discussion as to whether that's ethically or morally the right thing to do. So subsequent to this, there's been a recent review that David Beard has written on this to summarize again the range of placebo sham surgeries that should be done now across the pain piece to really confirm whether many of these things that we're doing have, again, mechanistic efficacy, or is it through, again, an expectation manipulation that they're producing the outcomes, if they're producing any outcome at all. And so it's going to be very exciting, I think, in the next 5 years as we start to do due process through many of these things and find out actually what's going on. So I wanted to share that story with you. Another belief sort of one is, again, slightly weird and wonderful one. This is one we did many years ago looking at religious belief. But again, it gets into these issues of rethinking pain, reframing pain, harnessing brain mechanisms that allow you to use something that's important to you and comforting to you, that allows you to emotionally detach from something that is suffering like pain. And for some people, religion is that, or it could be some other particular thing that's been a lifelong positive influence for them. We happen to pick religion. And in this study, we had a very clear hypothesis about a part of the brain in the front here that we know is very important for, again, detaching yourself from the emotional significance of things, and very easily accessed by people who are very good at being more of a, what we call an external locus of control, a person who's got more of a sort of disposition of letting fate just decide what happens and not wanting to control things so much. So the people who are very controlling aren't very good at activating this. And we know that in situations where we manipulate their sense of control of their pain, or in this situation where, again, they're maybe not religious, and generally religious people are more fate and more external locus control, able to show that the more you can activate this region, the more you can detach from the emotional significance and the less painful it is. And the galvanic skin response came down. And it was an extraordinary, successful experiment. Again, really media went nuts on this one too. But again, they lost track because they were focusing on the religion. You know, as I kept saying, you could take football fans and show a picture of David Beckham or whoever would be, you know, Marcus Rashford these days, and you know, and all our wonderful Mason Mount and all the guys that will be hopefully winning the Euros for us. You know, something that's been a very positive thing that you look at and brings, again, a sense of positivity. We just happened to pick this because it was actually quite easy to control. But it speaks to, again, things that, you know, patients, you know, find and have found a source of comfort and find that it can help. And what we're able to do with the science is just do experiments as best we can, just sort of go under the bonnet a bit and say, well, this is why this is doing, and this is real, this is physiology, this is brain networks, and this is what you're doing. And we can piece the jigsaw together, and hopefully that can provide new ways of thinking about it, again, particularly, I think, you know, psychological interventions for pain. Pain and pleasure. This is me and my daughter shamelessly showing off about running some marathons. This is me, London, and then many years later, the New York one. You can see my daughter just dragging me over the finishing line there. But these are, you know, again, these extreme events where afterwards, you know, you're aching like hell the next day, but it's nice pain because I'm just interpreting that whole body racked pain as good pain, because I've just finished a marathon and I'm associating it and reframing it as a good thing. Whereas obviously in chronic pain, it's not a good thing. But by learning how you can make pain pleasant and reframe it, and what are the brain networks that switch on when that happens, which is the types of experiments we're doing here, we're able to think, right, okay, if we can now get those bits of the brain on, that's again free analgesia. That's another way we can help patients again access some of these incredibly powerful brain networks to again bring a reframing of it and a reinterpretation of it. And again, that might just provide enough relief until such time as we can, you know, really cure the disease of chronic pain. For patients who, I don't know if any patients out there have phantom limb pain, you know, this is a devastating condition. I just want just one slide just to show this work that, again, one of my postdocs who's now a full professor down at UCL, just wonderful researcher who worked with my team and another team who works on stroke and plasticity, where we were really looking at the whole concept of maladaptive plasticity and what is going on. You know, is it the case that this supposed, you know, shift in the representation of the missing limb is what's driving the pain and a mirror box will fix it? Well, we, in a series of papers, have sort of slightly overturned that view and saying actually it's not as simple as that. The brain is certainly bringing something to the equation, but actually this shift is not as big as we think. And in fact, in amputees, the good news story is even if you've not had a limb for 20, 30 years, the bit of the brain that normally would represent that limb is not missing or not able to be active. It's in fact completely well and totally active. In fact, you wouldn't be able to discriminate from controls. What it's doing, though, is not talking to the rest of the brain. So it's completely capable of being activated, but it's now become isolated. And the more isolated it is, the more pain the person is in. So this is how connected this bit of the brain is, having a chat, say, to other parts. And what we're finding is if it's really bad in its connectivity, the patients are in a lot of pain. Whereas those who can chat away to other bits of the brain are in less pain. Well, that prompted the idea, well, why don't we blast this group with our favorite little brain stimulator and get it talking? Let's drive this bit that we now know can work and is perfectly functional. Let's make it have a conversation with the brain. Let's see if we can take these people and make them into these people. So we did this with a sham intervention and a real intervention, and we got profound drops in pain, which we thought we'd get acutely, but do you know what? It lasted for several weeks. So again, we're building on this work now as, again, a new non-interventional way of, again, mechanistically learning from what we've learned and hoping to bring a different way of treating, again, this devastating pain for phantom limb patients. The final frontier, as I mentioned, was to try and understand that as you go from a baby, this just gives you a nice example. It's a 3D plastic printout. Rebecca Slater gave me these pictures of a baby's brain, and you can just see how it develops from adolescence through to adulthood. So this is why that German anatomist said babies don't feel pain, because it's not folded as much, but we now know is nonsense. But nonetheless, you can see this is one journey, isn't it, of your brain and your life's journey, and its bumps and its scrapes and all the different things that it's going to, you know, be bombarded with. It does shape it literally and metaphorically. So what we're realizing is, well, what is it, and why is it that we're seeing that this sort of combination seems to be the predisposing for people who develop chronic pain? What's going on? How can we capture that into brain networks? And what are the brain networks that seem to be important for maybe explaining who's the 1 in 5? And can we correct them? Can we, if there is a vulnerability developed, can we reverse that? If there are things that we can learn that make people resilient by looking at the 4 out of 5 who don't develop chronic pain, can we harness that and bring that back in? And again, that's a direction of travel going forward. But to do that, we have to sort of look at big brain data. And again, this principle would apply. I'm going to just skip over this for time, but this can apply also not just for developing chronic pain, but also not being able to benefit from treatments. So again, some of the work we've done has just shown that, again, if your networks for, say, reward and analgesics in your brains are not quite working as well, then the drug's got nothing to interact with to drive that reward. So again, a lot of the sort of what explains the vulnerability or holding people in chronic pain, it's important to also look at what might not work in terms of the systems that need to be able to work well for the drugs to do their job. And again, this gives us different targets to look at going forward. So some big studies that are ongoing at the moment, if you're not aware of, are these studies involving Biobank, which is this fantastic, huge cohort study that Britain has been undergoing. We're very, very good at cohort studies in the UK. It's been our big thing for, best part of 50, 60 years. This is the new big one that we've been doing over several years now. And the focus is to, characterize 500,000 people between 45 and 70 for their brains, their hearts and their livers. Imaging studies, genetic studies, lifestyle studies, everything can categorize so that we can understand heart disease, dementia, other conditions. And we're very fortunate that the money was released to allow 100,000 of that 500,000 to have their brains and their hearts imaged, already up to 40,000. Our lab that I used to run is driving this and doing all the analysis. This database of all these beautiful brains being collected, where we have the genetics, the lifestyle, are they smokers, are they not, what is their physical activity, et cetera, et cetera. We've got all this data. This is publicly now available. So scientists around the world can just pull out this data and start to ask really interesting questions. And of course, some of the things we want to pursue is not just on a much bigger scale than we've been able to do with just 20 patients or 30 patients. We can look at, tens of thousands of patients and say, right, this is how the brain is looking in this condition or that condition. This is what's common. This is what's different. And then we can look at people even before they've got chronic pain and start to say, well, what is it about them that might be predictive? And so again, that's the direction of travel that we're undergoing. So we hope that going forward, we'll have better biomarkers from a genetics and environment, biopsychosocial, lifestyle, all the different types of imaging things that we can collect, other types of biochemistry. And then we're going to bring all this hopefully together in a more composite way, because it's not going to be, again, one biomarker is going to predict all of pain, because your pain is more complex than that. We're going to have to piece together all these different bits and bobs so that for one individual, we can almost in a Rubik's Cube type way, work out what's the combination for them that explains their pain and then is going to be predictive of what treatment is going to be best for them and most effective. And that's the sort of next part of the journey. So this is the most important slide of all. This is the slide where I get to thank the amazing team I've had the absolute privilege of working with over the years, not just the team that I work with from the imaging center, but my own pain team and my collaborators. You can see we have a lot of fun. It's a very international team. That is the nature of science, and people come from all around Europe and the world, and you get to become great friends with these people and continue with their journeys and their lives, which is, I think, one of the greatest joys of being an academic and a scientist is that incredible exposure and experience you have to all these wonderful, wonderful people and sharing their lives and then going off and, again, building their teams and all just to hopefully give you encouragement, all of these amazing young people, dedicated to understanding pain, to serving you, to trying to come up with better ways of understanding it and to treat it. So that's a very important slide for me to show you. These are the list of the names. I just want to acknowledge again all the funding agencies that support the work over the years and have been very generous in that. And of course, all the volunteers and the patients who've participated in that, and many, many collaborators too. So with that, I think I'll stop screen sharing, and I'd be delighted to try and answer any questions that you might have.
That's brilliant. Thank you so much, Irene. That was just so fascinating, and that's an awful lot of information in a short time. I know my brain's feeling quite full right now. Absolutely amazing, though. I mean, those pictures are just fascinating, aren't they? Especially the brain at rest. that's,
Yeah,
I mean, it isn't at rest, is it? It's just constantly working.
exactly.
We do have a lot of questions. I think for footsteps, the most important one that has been asked was from Leslie, who asks, the information you shared was fascinating, but I would be interested in the practical advice we can take from right now. So with all your experience and knowledge, if you were a patient with chronic pain, what self-help strategies would you use?
Yeah, it's a terrific question. I think what I hope you've learned is just how powerful your brain is in controlling what pain you experience, both for the good and for the bad. So, I've given you examples of where you can make it worse by being, with mood changes and being anxious, by over-attending, and then how you can make it better by distracting, by lifting your mood, and again, finding reward and joys and things. So to be empowered, hopefully, to know that that isn't sort of just fake, that isn't just masking things, that if you actively pursue it will fundamentally change real network activity, real neurochemicals in your brain, real physiology. And that itself is going to, not only bring hopefully relief or less pain, but what we don't know, but what we would suspect is that continuation would help again reset some of these hardwired negative aspects on. So keep at it. So think of the things that for you, bring relief, again, through those distraction type elements or those mood type elements, and then think of the things that make it worse, and then start stopping those and doing more of the other, and keep at it.
Brilliant. Yeah, I mean, this sort of follows on with Claire's question, which is, in your experience, do people recover from chronic pain? Recovery in this instance being pain-free from some or most symptoms.
Yeah, it's a great, I mean, as you know, many of the treatments that are, particularly pharmacological ones, are not effective for all, and even if they are, it's a little bit. So, sadly, the bleak situation is, there isn't total relief for many different types of chronic pain patients. For some of those, that say have more nociceptive inflammatory, and let's take the joint replacement. So, for a good chunk of those, that is. So that person could be in pain for 10, 15 years, then have a new joint. And if they're not in that group that I showed, have this sort of slightly more complicated pain to it, they'll be better. But the situation is, I think, unfortunate, the case that it's not common to see that. But I am very optimistic, because this shift in our thinking about chronic pain as a disease, and this shift in where we've paid our attention, if you like, in our science, what is coming through the pipeline, honestly, guys, in terms of new discovery. I mean, I'm obviously focusing on the brain because that's my area, but, other people come and talk to you about what the discoveries are at the first nerve coming in, the transduction, the transmission, and the new drugs and therapies coming through the pipeline. And it's not just pharmacology, the whole neurotechnologies, the whole monitoring and intervention. This is very exciting, I think. So I think the next decade is going to bring some really exciting delivery in terms of therapies on that new science that we've discovered, and that we're reframing again, that we've got to fix the disease of chronic pain, not just sort of, again, just, not window dressing, but just sort of, suppressing things. We've got to go in there and actually fix it, and then the analgesia will come.
Yeah, absolutely. I mean, full disclosure, I actually have recovered from chronic pain. I have to work at it, as you say, with the, with the psychological techniques, and making sure I do the good stuff. And it can be a part-time job just doing that. But I mean, I think what you're saying, though, is that this is a multidisciplinary thing for patients, and they are included themselves in the team, that they have to take on their own role and their own responsibility.
I think that's right, because, as I mentioned, generally it's the whole 3 neuron system that goes wrong. So certain things are going to be, needing, agents or interventions to fix it. And then, but the brain bit, you've got a lot of control on that bit of it. So that's sort of where you can come to the table with, again, but you've got to sort of work out what works for you. But hopefully, at least today, I've given you a confidence that it's there for the taking and the accessing, and the trick is to sort of, for you, find the keys to unlock those incredible systems in your brain to help you.
And they're different for everybody is the thing as well. We don't know. Ashley mentions the pain protectometer, the DIMS and SIMS, danger in me versus safety in me concept, which I think is Lorimer Moseley and David Butler. She said it's like knowing the good cop, bad cop strategies that are unique to you. So it's getting this information across to people, though, isn't it? And I mean, do you have any sort of suggestions for clinicians how they do this in the most effective way? Or
well, for those out there who are, say, working on, obviously from what I've spoken largely about today, you know, in terms of the brain-based mechanisms, will probably naturally sit more with anybody out there who's, say, working more on the CBT related sides of things. But I think for any clinician, irrespective of the background, I think just knowing how, again, powerful the brain element is in terms of making it worse and making it better, and engaging with that, and not being just unilateral in the focus of where the therapy is, because, you know, you could have a very effective therapy, but if you're not unblocking some of the more mood related problems and you know, cognitive problems, that therapy, say, at the periphery, is not going to do as much as it could be doing, or anything, because I've just shown you I can completely mask one of the most effective analgesics we've ever discovered. I can just make it look like it's not working at all. So it's that sort of level of just awareness of the conversation, again, that patient physician consultation, you know, and being mindful of how powerful that setting up of expectations can be, and real outcomes, I think, is something to just, you know, not to frighten them, but just be aware of, and then to use it and harness it. But, you know, to keep an eye, again, you know, on the fact that, you know, the three-pronged approach, you know, is going to be the way to go, and that there's going to be some new therapies coming out, and to make sure, you know, to engage with them and be part of the research, be part of the journey with us. It's the only way we're all together, you know, help really bring some, you know, delivery in terms of relief. Yes, I mean, I saw just very brief in the chat, somebody asked a question about the sort of, can you rewire your brain, you know, and again, that sort of repeated effort to try and use some of these systems. And the answer is actually, so it's not in the pain field yet being shown, but in the stroke field, where patients, say, who've had a stroke and they've lost, say, motor function, some people naturally, their brains are quite plastic, and they recover motor function, and you can map that their brain is recovering, both those neurochemicals and the wiring and the function. Other patients, not so. So what did the guys do? Well, they brought them in a combination of physical therapy with, again, the zapping, help drive them a little bit more. So those who needed a little bit of help with the plasticity got it with a combined little bit of a tickle of the brain with the physical therapy together, just pushes them a little bit quicker into driving the plasticity and driving a better outcome. So again, a lovely trial in stroke showing, yes, you can change. And then of course we can image and watch, you're changing the wiring, you're changing the neurochemistry. And if you do it longitudinally, you can make that brain that's become sick recover it back. Because the beautiful thing about the human brain, even if you're quite old, is it's fantastically plastic. So you can make it, you know, go bad, and you can bring it back again from the brain. That's what's really positive about it. And again, that's been shown time and time again with the different ways we look at the human brain, that the ability for the brain to recover and replasticize in a good way is absolutely there. Again, the trick is what's going to work for you best. And I think these combinations of cognitive behavioral therapy, pharmacology, physical therapy, maybe with a little bit of tickling of the brain just to help it, is how we're going to really, you know, get these networks working for you.
Yeah, absolutely. It's fascinating. It, again, shows the multidisciplinary nature of this. Yeah, I mean, I deconditioned all my triggers with graded exposure combined with positive reinforcement, because I'm an animal behaviorist person, so I use that aspect of doing things. So again, it comes down to knowing how you learn.
Yeah,
which was fascinating. Very
yeah, no, learning is a very, I mean, I had it on one of my slides, but didn't go into too much, but again, learning is very important. And again, it's all part of that, you know, when you're learning expectations and associations, like that patient I had picking up and loading the dice, you know, that every time you stand up, it's a learned phenomenon. You know, in a good way, you know, pain is a teacher, you know, so you learn to avoid it, but you can't avoid it if you're a chronic pain patient. So again, that learning becomes now aberrant and you know, and again, that can drive other things. So again, learning is another really interesting area that's being looked at, and again, it's an opportunity, I think, for again unlearning negative associations and relearning good ones. So again, getting those learning networks and mechanisms going.
Somebody asked in the chat if these brain imaging trials are done on people, are they done on healthy volunteers, or I mean, are you doing them on people with chronic pain already to see how their brains are different, react differently?
Absolutely, yeah. So what generally the approach is some of the basic discovery stuff you'll do in healthy volunteers, and then in what we call experimental medicine models, where we have certain, obviously we're limited, we can't make healthy volunteers chronic pain patients, but there's ways that we can simulate some of your symptoms. So we can put chili pepper cream on, and that just is ongoing burning pain. It then can create hyperalgesia and central sensitization, and then we can wipe it off and it reverses. So we've got certain ways, and there's other tools that we can simulate some of the mechanisms that are important and some of the symptoms. And then what we do is then, having discovered the science, we'll then go into the patient cohorts and we'll verify it there. So my talk sort of interleaved between the two. If there was a sort of, if I said sort of discovery basic mechanism, that was often in a healthy control, and then you'll see like the OA and the diabetic painful neuropathy and RA. So I was showing you then the different pain conditions where you then prove that this thing is now in the patients and this is causing more or less of their pain. Yeah, absolutely.
Yeah. There's sort of, on your slide, you put the three different types of pain, the nociceptive, neuropathic, and you called it idiopathic, functional. They also call that nociplastic, don't they? What's your view on that?
Yeah, I mean, it's always good to have new discussions around terminology and how we label things, because labels become important, you know, in a whole race, and also it helps, I think, in the science, in terms of, again, saying this is the mechanism we think is important, and then we want to see if that thing is present in one condition or all conditions. So this whole sort of, you know, nociplastic thing, I mean, it's I mean, at one level, you know, one could say for a lot of biology, it's the same. It's just you've got different semantics in terms of how you do it. I mean, there is no doubt, you know, we're very clear about, again, a healthy nociceptive system, and how in the transition to persistent chronic pain, things become aberrant right from that transduction, transmission, and things and perception level, and how things that are maybe of a more reflecting an amplification state, and whether that's peripheral or central. So again, a lot of these terms in terms of, you know, have you got a more peripheral nociceptive driven thing, or have you gone a little bit more nociplastic where there's more complication to your pain? So I might use different jargon to explain that, but, you know, again, it is helpful every now and again to sort of pull maybe a few different concepts into, again, more simple bins in order to just put a label on it. But when you strip it back, you know, often one's coming back to sort of same principles of what you're discovering. And I think, you know, fundamentally, I keep it really simple. You know, it's all of perception is a three, you know, neuron system. They all go wrong, but people's chronic pain can be heavily more peripherally driven or more centrally driven. Generally, it's a combination of the two, but it might be more weighted to one or the other. But either way, you need to fix it all. And if you do stop it coming in, things will calm down centrally. But sometimes the central is really dominant. So you've got to calm it down there and then calm it down the periphery. So again, that balance between nociceptive, inflammatory versus nociplastic, more functional, it's a spectrum. And everybody's somewhere on that spectrum, the mixture, but the weighting might be a bit more, you know, central, a bit more peripheral.
How hard is it for people who can't stick people into scanners to determine if somebody has a large sort of centralized aspect?
Yeah, so my view about sort of the types of stuff I do, you know, is very much in the research domain, it's in the discovery domain, and we look at patients, but to answer the scientific question and prove the point. There are several examples where then, when we've done that, we say, fine, the scanning has done its job because it's verified and proved the point. Now what we need to do is work out what would be a more simple bedside measure. Could be a questionnaire. It could be using, say, a simple quantitative sensory testing, poking and prodding and getting thresholds, but that is reflective of what we've discovered in the brain and tells you something about, are they going to respond well to this particular treatment? Do they have more central pain versus peripheral? So some of the examples I've given you, and again, just for time, even though I went on for 2 hours, I didn't show you, but some of those examples I've given you is exactly what we've done. It's like the imaging's done its job. You're not going to have the imaging rolled out, certainly not on the NHS, not with all those fancy sequences that we have easily, and neither should you. It's too expensive, but it's done its job now, and now it's our job to say, right, well, what would be having discovered something you didn't expect, what would be a different questionnaire that you might not have ever thought to use in the clinic, but could be really revealing in terms of who's likely to respond to this treatment or that. So it's called reverse engineering, from the bench to the bedside, and then being informed by something you might have discovered, saying, well, okay, we're not going to roll the imaging out, but the imaging told us that this network's really important, and we wouldn't have thought about that before. So why don't we go and find out there? Because often in the literature, there's always something. What technique that's simple, what questionnaire that you never thought to use actually could be really revealing for, again, not just helping understand the patient's pain, but importantly, predicting which treatment is likely to be more effective. Because until we get all these new exciting drugs through, we've just got to use what we've got better. So, that's currently what we can do right now is just say, what can we be doing better by not having you go on this treatment and that treatment and that treatment, failing. What is it that we should be just doing better to do that? So part of these big, funded platforms will start to develop more, I hope, efficient, streamlined bedside measures that, ultimately, hopefully can be rolled out, at a GP level, such that it could be more effective for putting you on the right treatment and not putting you on the wrong treatment that already we're saying this is unlikely to work with that particular constellation of features that you've got. So there's just no point going on that one and wasting 3 months till you see whether it works or fails. We're not there yet, but that's a lot of where the effort is going into. So let's use our current things better and use what we're discovering to stratify or personalize medicine, whilst in the pipeline we've got some new things coming out treating the disease of chronic pain.
There are quite a few people in the chat commenting that chronic pain needs to be addressed a lot sooner in primary care. Yes. and given better thing. I assume you agree with that.
Absolutely. Yeah, it's great questions, everybody. It's really terrific. Yeah.
Susan asks if a physician's bedside manner, e.g. empathy, compassion or disinterest, can actually influence the success or otherwise of the treatment therapy they're prescribing.
Well, it's a great question. I mean, I would say yes, because of, again, what I've shown you about, again, managing expectations. Some other work that the question I might want to look up is by Karin Jensen. She's in Sweden, Jensen, or I think it's Sen. And she's done some very fascinating work, again, using brain imaging, but other measures too, to look at how, again, even if the physician doesn't know what treatment the patient's on, or does know, how those subtle clues can be picked up and change the outcome. So again, this is important in an extreme case in sort of randomized controlled trials where nobody knows what's going on, but also obviously in these interactions of these social contracts that we get into, that subtle things that even a physician might not be aware of that they're giving off because they have an expectancy or an attitude about a particular treatment will influence. So she's done these quite sophisticated scenarios of different types of, again, patient-physician interactions and what cues are being picked up, even implicitly, without the physician being aware. So I urge you to read some of her work, which again speaks to, I think, just how impressive we are as animals and humans at picking up clues from each other. I mean, haven't we learned a lot from the Zoom for the past year that it's a very transactional interaction, isn't it, and meeting, and what we've missed in that subtlety of picking up cues on people. It'd be very interesting, actually, because of a lot of I'm interested if any physician out there or somebody who's done a study to know whether, again, how the outcomes on patients of treatments by doing remote clinics through Zoom would be different to the in-person, because that subtlety of that exchange will be missed on the Zoom. Maybe might be better.
Yeah, it's certainly one to, I hope people are doing some research on. There's a lot of opportunities at the moment, aren't there?
There certainly are.
How are we doing for time? Are you all right to answer another one? Or
yeah, absolutely. Yeah, no, I'm happy as long as people are here and want questions. I'm very.
Oh, we might be here all night at this rate. Tim asks something you were talking about. You were saying about recent brain theory showing the level of activity that exists even when there's no external stimuli. You referred, I think, to a model or reality. Is this predictive? Does the brain run a program calls it sapience 2, that interacts with external reality.
Oh,
It's
wow.
an interesting
I'm
one,
not sure I
isn't it?
fully understand the question. Yeah, I'm not sure I fully understand the question, but from what I've heard, Nikki, from what you've described, so it sounds like the person is asking whether there's a sort of internal program that you're just going through this and this. I don't think, well, the answer is we don't know. I mean, this new way we're thinking about the brain working, and this recognition that there are these priors and how and where they produce that influence on the bottom-up inputs is very early days, early doors. So only really, the past few years are we starting to get a better understanding of it, and not only the power they have, but where do they produce? How are these priors actually influencing, let's say, vision? Where is it turning the visual cortex up and down? What is it doing? Where is it? Or is it very high order, as we say? And so some basic questions are still not understood about that. But in terms of, again, a programming, obviously, if you're going to be, generally, there is a moment in time when you're having that window of expectancy about what you're going to see or hear. And that's what's going to sort of flick in that network then to be ready to go to sort of, okay, I'm gonna be listening to this music now, and I'm expecting that to be nice. And so again, there'll be things that will just come at you very directly, and then maybe there does have to be a fast shuffling system. And then other experiences, you'll have a bit of time where, again, like I was saying, picking up the glass, that gives me a window to say I'm going to pick up the glass. That's a very simple one where that's going to just go straight away. The interesting one will be where it's more fast, and whether even in those faster situations the priors can not have as big an effect. Don't know.
It's fascinating. We've got so much to learn still, haven't we?
Yeah. Well, I think that's the important thing, I guess, to emphasize, which I haven't said, is, by definition, I'm showing you the new theory of how we think the brain works. That in 10 years, I can come back and say, well, that was wrong. We've got this new theory now. And so, we're trying to understand this really complex thing, pain, which we still haven't, got a good handle on, but we're getting there. And at the same time, in parallel, we're fundamentally trying to understand how the brain just works anyway. So every time we sort of update our model of the brain, we sort of rethink, and it's fun, actually. Sometimes I go back to this experiments we did, go back to, say, some of our very early experiments 20 odd years ago. And now I would reframe that in our new understanding of the brain in terms of priors and expectancies. Whereas back then, we interpreted it in a certain way. So that's, again, part of the evolution of science, but particularly I think in neuroscience, where we're not working, unfortunately, from a preferential position of knowing well how the thing works. It's not like we're sort of working on heart disease, where you've got the absolute basics of how the heart works and pumps things around. We're not in that privileged position. So that makes it even harder. I'm not giving excuses. I'm just explaining why the progress maybe isn't as quick as you hope.
No, it's still been astonishingly fast as far as pain's concerned over the last 15 years. I mean, it's really made a big difference. Yeah. Francis is asking about sleep and the brain. Do we know what's happening within the brain that we can change?
Yeah, sleep is a great one. We have in our department some terrific sleep researchers and people who work on circadian rhythms. And I often joke and say, if I was going to start my science again, I'd definitely get more into sleep, because it's such an interesting. I mean, we spend most of our lives asleep, but obviously it goes wrong in many conditions, and it absolutely is one of the comorbid issues with pain. And there's some really interesting interactions. So what we know about the sleep systems, and again the circadian rhythm controls, all center on some of these old brain networks that, again, interestingly overlap, with, again, some of those core descending systems. So suddenly you can start to see, and there's been studies done where people get sleep deprived, healthy volunteers. If you sleep deprive them, they can produce almost a fibromyalgic like hyperalgesic state, and we can see that the brain stems start to go crackers. So there's something there that, again, is interrelation. There's also some really exciting stuff looking at, again, circadian rhythms and disturbance there. And these will be, again, we're in fact going to start a trial looking at sleep intervention using one of these sleep therapies in patients with fibromyalgia as an intervention to, again, hopefully help with the sort of cognitive fibro fog, but also to have a benefit on the pain too. So I'm a big fan. So it's a great question. So thank you for asking it. That there's there's a lot that we need and could be exploiting more around sleep and its interaction with pain, both at a mechanistic level, but absolutely at an intervention level. So thank you for the question. Yeah.
Brilliant. It, again, emphasizes how many things we can do to help the experience we have of pain.
Yeah, absolutely. I mean, good sleep, you know, the sequelae of having good sleep in terms of mood changes, and again, absolutely lots of profound neurochemical changes, calming down, lots of excitatory neurotransmitters, better physical health. It's profound, and these will have absolutely good effects. So you know, getting into good sleep habits and again, what works for you, very, very important.
It's this diet as well, and exercise, movement, all those good things.
That's right. That's right.
It's a very empowering message, really, and one we're trying to push, you know, at the festival, that there's a lot you can do to help yourself. But it's certainly very good to see how much is being done with you guys, and you know, the things that are going to sort of add on to that, and hopefully, you know, make those things even more effective. We talked a little bit the other day about our new discovery zone at the festival, which is a new zone on the main stage, which is introducing people to research into pain. I'm not sure if you've had a chance to look at it, but we've got some really good videos there on already, and the Cochrane review will be on there soon. So I really encourage everybody to have a look at that, because being able to access research is fundamental, isn't it? It's a really important thing for people.
Yeah, and we've spoken about maybe getting some of our young guys, you know, to take a research paper and you know, explain it, you know, and give people the sense of the journey of, you know, I think it, you know, it's just one, it's good to meet the young people who are working, you know, in the space, and that's you know, hopefully invigorating and encouraging, but also it's good for them to be able to communicate again the science, but also it gives you a sense of just, you know, how not just obviously understand what the science is telling you, but just the complexity and just why it takes so long, because of just, you know, I think if you sort of get a little bit in the nitty gritty, it sort of just gives you a better sense of sort of, again, the complexity of you know, these experiments and the incremental nature, and how rare it is to have a major, big, profound discovery, but you know, a good sense of that journey, so that you know, again, you can be part of, also, you know, part of joining it, you know. I mean, there's so much now research, thankfully, funding coming into the pain space, which is really great, and much of this, and many of us, you know, in terms of PPI and engagement, this is essential. You know, we need to be asking the right questions that are addressing the issues that, you know, you really want addressing, and we can only do that if we're all in partnership together. So I think, you know, getting through your platform and the discovery, you know, more exposure to some of the scientists doing it, it's a win-win on all sides, I think. So we'll have to follow up with that, Nikki and Louise.
Yes, definitely. It's all very exciting. I think there's just a few comments on the new NICE guidelines. Are you minded to make any comments about that? Somebody specifically picked up your point about the pregabalin and the excitability.
So is there a particular question they want about the pregabalin, or? Well,
the NICE guidelines obviously have suggested that pregabalin shouldn't be used for that. So I think they're querying.
Oh, I see.
Why, you know, if that's actually what you think of that.
Yeah. Oh, okay. Yeah. I mean, it's without going into specifics on that. I mean, even if when you saw that study, you saw that for some people had a high. So it was those who had the very high one brought it down. So again, even within that, again, when one's looking at average effects, even if if I put the slide back up again, or maybe just go and have a look at the study, Harris, Anesthesiology 2013, you know, and Harris would be, you know, I can get him to come and give a talk on that one. You'll see that there's an array for some, you know, that wasn't aberrant and that isn't going to have an effect. And for those that it was, that's where the most effective effect was. So again, when NICE comes up with their guidelines, it's what's going to maximize the benefit for all. And again, so I think that probably best explains that particular one around the pregabalin, particularly in that condition.
And obviously, drugs are not without side effects and their own
Well,
negative
that's
problems.
exactly right. Yeah, exactly. So it's always it's a cost balance, but on monetary terms. It's about effective and side effect.
Nancy asked if you know of anybody who's working on anti-inflammatories, specifically pain medications.
Yeah, so there's a lot of just the whole sort of neuroinflammatory, inflammatory, and then I extrapolate that to say neuroimmune. There's a lot of interest around, again, some of these mechanisms that maybe we haven't looked at specifically. And I think if you take, say, rheumatoid arthritis and sort of anti-TNF therapies, there's a lot of developments that are broadening out the opportunities to target inflammatory mechanisms that have gone aberrant with different new therapies. So it's actually quite an active area, not just in the pain field, actually, just more generally, because we're recognizing that, particularly in the central nervous system, again, some of those changes that are not just neuroinflammatory, but also neuroimmune are things that, again, maybe druggable, maybe targetable, and there could be new opportunities there. So I think this is a very interesting and good one, because, again, you could, even in conditions where they maybe would have been thought of looking for where the nerve injury is, but it could be that, that's not been maybe easy to find. And again, it could be that there's a different mechanism for it. There's more around, again, an inflammatory or a neuroimmune problem, and that's more where one needs to go. So yes, the short answer is there's plenty of work going on, both at a basic science level and, again, at a sort of pharmaceutical industry level around that broad space, and not just for pain. And often, as these core systems, quite often, again, that the body's efficient. You're not going to have a separate system for the pain system as you're going to have for other systems. When they go wrong often in several things. So sometimes one can learn from other diseases and disorders that drug that's really effective for that mechanism, that's a real problem, that one actually can have really good effect for a subset of patients where that's a big dominant component for their pain. So it's about making sure that people are talking to each other across these spaces, in core mechanisms, rather than always getting siloed in your own disease area.
Yeah, I agree so much about that. Medicine can be very reductive, can't it? And we forget to look at us as a whole. I mean, you go to 3 different consultants about one problem, basically, or to you it's one problem.
Yeah.
I think the last question, I think, because you've been absolutely brilliant staying so long.
No problem.
Somebody, this is Dr. Brian Geldry, who's asking if you research alternative therapies with your on your brain image.
Oh yeah, it's a great
things like he talks about the Alexander technique and acupuncture. Somebody else in the questions mentioned hypnosis. So all these interesting ones.
Yeah, no, there has been work in imaging done both in acupuncture, hypnosis, mindfulness, meditation, and I think these alternative therapies or CAM, and there's a whole NIH-funded center for complementary alternative medicine looking in the context of pain, and indeed showing, again, with objective evidence that this, it's not got the clarity yet for all these different ones, how it's working, but certainly it's doing something and it's changing, things. The foray that we've made is with mindfulness, with irritable bowel syndrome, where we were opportunistically able to bolt onto a trial that was happening anyway, clinically, and we just added an imaging bit to it. And that's been really, really interesting in terms of, again, looking at how those interventions are producing changes in, again, you can almost see the sort of how the network becomes sort of dislocated, if you like, in a way that you would sort of predict in terms of, again, taking off a sort of attention network and refocusing it. So we're still, my student who did this as part of her doctorate, she's then got off on maternity leave and had a lovely daughter. So she's just coming back now, finishing her surgical training. So we're still analysing the data, but that's the closest we've personally gone. And there's certainly some really interesting things, I think, for us to explore in that context of mindfulness. And I'm interested in that because it's sort of, there's a spectrum, if you like, mindfulness, meditation, hypnosis. And again, we do this work that's a separate sort of arm of my research on mechanisms of anesthesia and how they produce. And that's a very extreme end manipulation. So I'm very interested in these techniques more and more. So we will be doing more of that type of work. But prior to my work, there's been quite a healthy amount of work being done in that space, both in terms of, again, acupuncture, hypnosis, meditation, mindfulness, even things like taking, people who are, yogis and have done a lot and look at their structural brains in different regions. And again, how they respond to tonic, cold, stresses versus people who are not. And again, how they are able to sort of activate some of these, again, core systems that just keep the pain down. So I think they're really, I think the imaging lends itself very well for those studies that maybe people find hard to get traction because of the sort of medical biases about, it's got to be drugs and that's it. And in the absence of actually seeing what's going on, what's good about, our technique is a picture tells 1,000 words, and it's hard to argue when you can just see, well, that person's brain doesn't look right. Okay, so stop saying they don't have pain when it's looking completely, not right, compared to, an age and sexual control. And don't tell me this thing's not working when I can show you it's suppressed things and there's less activity. So it's very powerful for, again, just getting the conversation moved on. So yeah, hope we can do more of those types of studies.
Yeah, that's brilliant. I'm going to sneak one in on my own regarding your surgery and the placebo surgery. Does it work if it's an open placebo surgery? So if they know it's a placebo surgery? Don't know.
Great question. Certainly the only study that's close to that is Ted Kapchuk, who does a lot of placebo studies at Harvard. You probably know the study. He did an IBS open placebo, where he told them this is a placebo pill, it's got no active ingredient. But, these are the studies that show if you take it and you expect it to work, you can drive some of these systems. And they got still a significant effect. It wasn't as much as the full Monty placebo, but it nonetheless. So again, if it's done well and the expectations are managed well, there would be what I'd predict, but I don't know of any data that's done it.
It's
great question.
An interesting one to do in the future, won't it?
Indeed, yeah, absolutely.
Because obviously there's ethical questions regarding placebos and not telling people, which I understand, but also it's a bit frustrating at the same time.
Yeah, absolutely.
Okay.
Yeah.
But anyway, we'll wind up now. Thank you so much. I can't tell you how brilliant this has been. I absolutely loved it, and I think everybody else has as well. And thank you so much for everything.
You're very welcome. Well, I'm happy. I'm sorry I'm not able to answer all the questions, but happy to sort of, you can save the chat if you want, guys, and I can post on your site answers to other questions I've not done. If you want to save the chat, then do so, and I can have a go.
That would be fantastic, because we can email anything to people afterwards.
Yeah, sure. Happy to do that. Yeah.
Oh, awesome. Thank you so much. I mean, I think some of the questions sort of you answered sort of during answering other questions. So hopefully I got most of them. But,
great. Cool. Well,